Okay, this is part 2 of your notes for DNA, RNA, and proteins. So underneath your notes from the first video, write down making a protein part 2, translation. We just went over transcription, which is the first part of making a protein. In transcription, we had DNA and we made RNA. In translation, we're going to take that mRNA and make proteins. Okay, so when I think of the word translation, it makes me think of what I do or what I see others do when they're translating languages. So it's basically the same thing that's happening. We're taking the mRNA and we're translating that information to make proteins. Similarly, I have translated for my parents from English to Spanish so they can understand. We're translating the mRNA to make a protein. So over transcription, we said it started in the nucleus and then it made its way out. That's what mRNA did. It was made in the nucleus, and that's why transcription is circled for the nucleus, and then it makes its way out. Translation only happens in the nucleus, outside of the nucleus, where you see the red circle labeled translation. Notice it's circled above ribosomes. That's important. Translation involves ribosomes. So practicing ribosomes, because we're going to say that a lot. And then, you have your plasmid membrane and everything else in the cell. Your ribosomes are not in the nucleus. Sometimes they can. Point is, ribosomes, where it's circled right now is your cytoplasm. Transcription is circled for your nucleus. Okay, so look at this little GIF and think about what's happening. What do you see? What do you notice happening here? So I see this little pink strip with the hat, with the baseball cap. He's mRNA. And that's sliding through a little purple person labeled rRNA. So mRNA is going through rRNA. So M goes through R. Okay? And then I see these little blue guys labeled tRNA. The little blue guys are holding like balls and they're attaching them together. And they're just like sitting on top of this mRNA. So it's actually what happens basically. So there's 3 types of RNA that we're looking for. mRNA, which stands for messenger. We already went over that in transcription. Then there's also tRNA, which is T for transfer, and rRNA, which is ribosomal. So if we look back at our little picture here, mRNA is the messenger RNA. rRNA is the ribosomal RNA. And tRNA is transfer RNA. Okay. Messenger RNA carries the code, where tRNA, transfer RNA, breaks the code down. And rRNA, the ribosomal RNA, is the site for code breaking. Let's write that down. So there's a few steps for translation too. So translation, we have a red light and a green light. First, we have to get connected. So the ribosome attaches to mRNA and slides to find the start codon. So if we think back to our little GIF, the little purple guy, our mRNA has to slide through the purple guy, and we don't start adding those circles until we hit start. So we have to have the green light to start. So if we look at this picture that says codon and it's circling these 3 letters, that is your start codon. AUG will always be a start codon. Like, you need to know AUG equals start codon. Write that down. The start codon, AUG, produces an amino acid. The amino acid is methionine. That's why it's met up top. So mRNA is what we made in transcription. tRNA is the red square that's breeding and making the anticodon. And then amino acid is being produced. Okay. And a codon is a fancy word for a group of 3 mRNA bases. So we already run over our bases. A goes with U, C goes with G for RNAs, right? So 3 mRNA bases is a codon. Write that down. 1 codon codes for 1 amino acid. And this picture is showing methionine as our amino acid. We have 3 letters at the bottom, making 3 letters that match it, making 1 amino acid. And after we get our amino acid, we're making a chain. So if we look back to the picture, our amino acids are those balls that the blue guys are holding. Each little ball is an amino acid, and the tRNA is attaching them to each other. The ribosome slides to the next codon and brings in the next tRNA. The amino acids bond in a chain and they repeat. So that's why this GIF is like literally never ending, because it's never ending until you hit the stop. Oh, look at that, it's the stop. Stop it. So the third step is the stop. Okay? Same thing, we needed a start codon to start it. We need a stop codon to stop it. Literally no fancy names. Start codon, stop codon. So the three letters, they're listed in the little graphic there. UAG, UAA, and UGA. You don't need to memorize the codon specifically. You will have a wheel that tells you. So it's basically a key. You're going to get a key. So as long as you know, we need a start codon to start, and we need a stop codon to stop. That's what you need to know. And ribosomes go until it sees a stop codon, ribosomes release, and the proteins fold. They fold. That looks important. So if it's bold, write it down. Super duper important. Let's go back. So first we have to make our codons. We get the start codon and we make it. Find your three bases. That's how you make one. Make your amino acids. Once you have your amino acids, you put them in a chain. You put them together. Once you put them together, you're going to hit a stop. And you have to stop. And then you produce your proteins and you got to fold it. So here's some more science jokes for you guys. So we have a man and Mr. Bean is cheating off of him. And then the one on the right says, "Can I copy your homework?" And that's RNA. And DNA replies, "Yeah, just change it up a bit so it doesn't look obvious you copied." And RNA says, "Okay." So I'm not sure if you got the joke or not. If you know the joke, good for you. If you don't, come see me and I will tell you the joke. Because you should understand the joke. Okay? And now this is just a practice run. Basically, transcribe the DNA. So this is your DNA. Transcribe. I want you to give me what the RNA would be. So think back to your rules. What would go with A for RNA? What would go with T for RNA? What would go with C for RNA? And what would go for G for RNA? Okay? And now we're going to practice translation of mRNA. So the first line of those letters, that's the DNA I gave you. The bottom line, that's the mRNA. You can tell because there's U's instead of T's. Because there's U's in RNA and T's in DNA. This fancy thing is called a codon chart. Codon wheel, whatever floats your boat. What's a codon? 3 bases. That's why we start with the middle one, work our way out of the circle. So the first one was AUG. So I'm going to read it from the mRNA. I don't look at the DNA when I'm translating, right? So I'm reading this. UAG. So I'm going to start off with U, the center of the circle. Then I'm going to go to the second letter, second circle. So I'm going to go to the A. So UA. And then I'm going to go to the G. Which letter's G? At the third circle, right there. This says stop. So I wouldn't do anything there. The next set is CUG. So I'm going to start at the C because it's the first circle. Second circle is U. Which one's U? Right here. Third letter is G. Right here. So if I have CUG, I have leucine. Okay? And then you're going to do the same thing for the last one. U for the first circle, A for the second circle, and A for the third circle. And that's a stop. So if I stop, I stop. Okay? And this is just a nice picture for you to look at. So we're taking DNA, we're opening it up, and we're making mRNA, and that's transcription. And then we're going to translate that to make amino acids to fold up to make our proteins. A chain of amino acids is a polypeptide. Polypeptide is just the fancy term. If you want, you can use the fancy term polypeptide. If you tell me it's a chain of amino acids, that's the same thing. And then there's this little video for protein synthesis. Open up your notes so you can watch it. It's about 2 minutes long.